Please join me in welcoming to the Distinctive Voices podium, Dr. Sada.
Jennifer, thanks very much. Good evening, everyone, and thanks for being here this evening. I'm going to have just a few introductory remarks introducing our speaker and this lecture, the Stephen J. Ryan Memorial Lecture. So again, welcome. It's great to have you here this evening. I want to also make a few acknowledgments because this lecture is, I think, a wonderful collaboration and jointly sponsored by Doheny Eye Institute and the National Academies. And so thanks to Susan, Marty, Jennifer, and all of the wonderful folks here who put on this great program. I also want to thank Christopher Conway in our development office, who puts a lot of time and effort into making this program possible, and Janet DeMint, who's the program director for the Ryan Initiative for Macular Research. I'm just going to take a couple of minutes to talk about that as well, because that is the event that follows this program starting tomorrow. So a few other people to thank, though. We are very fortunate at Doheny Eye Institute to have wonderful, committed supporters of what we do at Doheny, and they're big supporters also of this Ryan Initiative for Macular Research. Folks like the Rinkers and the Gavins, and we're grateful to Paula and Louise Gavin for being here this evening. But so many wonderful folks and foundations who are vital to what we are able to achieve. And this initiative for macular research that begins tomorrow is one of the types of very important programs. So let me just take a couple of minutes to talk about the person in whom this lecture is being given in his memory, in Steve Ryan, because Steve was certainly a really important mentor and friend to me, a great visionary in our field of ophthalmology, really a giant in terms of his accomplishments. Sometimes people say, he wrote the textbook on that. In his case, he did. He was, it was Ryan's Retina, which is our chief textbook. He's had enormous research contributions that too many to talk about. He was a leader in so many of our different societies in our field. He's gotten so many awards from the most important organizations in our field as well. And of course, he was a very important contributor and leader in the National Academies. So it's really wonderful that we're here at this venue. It just makes perfect sense given Steve's role and influence there. And of course, he was a leader in many efforts, I'm going to introduce our speaker in a moment, and he certainly is well aware of Steve's great advocacy for vision science research, and Steve certainly recognized that was important to be able to move the needle in terms of actually improving our knowledge and understanding of eye diseases. And certainly last but not least, in terms of his leadership, one of the great things that Steve achieved was he was, of course, the key leader who I think was responsible for this great development and evolution of the Doheny Eye Institute into a global center of excellence. And of course, many of you know that more recently we've had this historic affiliation with UCLA and the Stein Eye Institute that's really brought together two real powerhouse institutions to create, I think, a wonderful collaboration, really a great center of excellence for the world. And this is actually even our new building in Pasadena for the Doheny Eye Institute that we are developing as an integrated place for clinical care, for research, as well as education. And that's a good segue to talk about Steve as an educator. He had so many residents and fellows that he trained. It was really important to him. In fact, something that he thought was the most important thing. And these people that he's trained really have gone on to become the leaders of other institutions around the world, and really reflects his great global footprint. So again, when I talk about this Ryan Initiative for Macular Research, and this lecture today is really the preamble for this meeting. Steve had this great vision that he thought that if you're going to accomplish big things, you needed big teams working collaboratively together. And so he thought, well, what's a big goal that we can achieve? He said, well, we don't have any cure for macular degeneration. I mean, it's something that affects so many of us. We don't have a treatment because it's hard. I mean, if it was easy, we would have a treatment. And so he thought if you're going to actually make real progress, you actually needed to assemble a critical mass of scientists to really study this disease, arm them with the appropriate resources, but not just any team. You needed to have a team that was interdisciplinary. So by that I mean, like, maybe you need to have some chemists, engineers, people from diverse backgrounds who may not know anything about the eye, but may have important things to contribute that actually could move the field forward. Maybe think outside the box. And he also recognized that it's great that we're able to move things forward in terms of science, but there could be a long time horizon between new discoveries and actually getting to treatment. So he said we need to also get these industry leaders and scientists from industry involved. And they also play an important role in this meeting. So as I said, it just made perfect sense, and so we really are grateful to the National Academies because they obviously have this wonderful Distinctive Voices series. And we've joined forces essentially in having this lecture as part of this series, but in honor of Steve Ryan. And this is a great tradition in preceding this meeting, which again will start tomorrow. So last year we had a wonderful lecture in the first annual Stephen J. Ryan Memorial Lecture in Emily Chu. It gives me great pleasure. I'm immensely delighted to introduce Emily's boss, who's our speaker today. Who's Paul Sieving, who almost needs no introduction, but Paul is our 2018 Stephen J. Ryan Memorial Lecturer. He is the director of the National Eye Institute at the National Institutes of Health. He's also a member of the National Academy, so again, very fitting that we are here. And actually, not only the National Academy is here in the US, but in Germany as well, which I thought was really pretty impressive. Paul, of course, has a very exceptional training pedigree, and you can see by looking at this that he's spent time on both coasts as well as in the Midwest. So I always thought of Paul as a Midwesterner because I actually knew him. He didn't know this, but I was at University of Michigan as an undergrad, and so he was. I think he was just starting there at that time. When I was finishing, but in any event, it's really quite amazing what he's accomplished and where he's been. In fact, he's got just an enormous list of awards, lectureships, and publications, over 200 invited lectures, more than 200 scientific papers. He's really been a leader in vision science research. Even before he became director of the National Eye Institute, he was obviously at the University of Michigan for around 16 years, and he founded the Center for Retinal and Macular Degenerations, as well as a molecular ophthalmic diagnostics laboratory, which was even CLIA certified. But now is a director of the National Eye Institute, and Paul has been in this role for about 17 years now, as you can see. He manages a budget, oversees a budget of over $700 million that supports all of this great basic translational and clinical research that happens across the country. So he really, is someone who inspires and makes this work happen, in addition to all the great stuff that he's doing in his own lab. He also, in 2013, launched a very important initiative, and this is just, again, this maybe very similar to kind of Steve Ryan's concept that, you need to sometimes establish these big goals, these audacious goals, initiatives to really try to develop treatments for retinal diseases through regenerative therapeutic strategies. He is really an international authority in human retinal neurodegenerative diseases. And in 2015, he actually initiated an ocular gene therapy trial for X-linked retinoschisis, and he really is the world's expert on this condition. I'm sure he's going to talk about that in his lecture that I think we're in for a real treat. So again, I'm incredibly delighted, and we're really grateful to Paul for being here and being our second annual and 2018 Stephen J. Ryan Memorial Lecturer. And he's going to be speaking to us about opportunities for ocular gene therapy. Thank you.
Well, it's nice to see some faces in the audience. Thank you for coming tonight. I'd like to spend a few minutes talking about gene therapy, and particularly ocular gene therapy for eye diseases. Vaz, thank you for that very generous introduction, and it was wonderful to see Steve Ryan's infectious smile Steve truly was a visionary in how to put together some of the pieces that add up ultimately to new treatments for people. I will now tell you my one joke. You can see that the National Eye Institute is in Washington, D.C., so I've got to say, hi, I'm from the government. I'm here to help you. Not sure how that's going to... My mic, I think it's on.
The podium mic is on, but...
Now it's certainly on. My second joke may sound like a joke, but it's not a joke. Thank you for paying your taxes. Because the National Eye Institute is part of the National Institutes of Health. And could you turn that volume down a bit, please? Thank you. It's part of the National Institutes of Health, NIH, which this year has a budget of about $34 billion that goes into medical research. And I think all of us understand the importance of that. We have friends who have cancer and are being treated with new CAR T therapies, et cetera, et cetera. So thinking about where the future is coming from, I'd like to spend the next few minutes talking about gene therapy. But for me as an eye doctor, it starts with seeing patients. These are the kinds of patients that I saw previously at the University of Michigan when I was medical faculty there. This child is not looking quite straight at you, is he? Or here, the lens of the eye, you can see the white speckles of a cataract. And if you look through the pupil, you can actually look at nerve tissue at the back of the eye. And it's not particularly easy to see, but an ophthalmologist will look here and say, your son or your daughter has a form of macular degeneration caused by a gene. And in fact, now we have about 600 genes individually in test tubes that when they are altered in any of us will cause eye disease. Last year, Emily Chu was here, and she is an expert on AMD, age-related macular degeneration, and you see the similarities here, the optic nerve and the blood vessels, here the optic nerve and the blood vessels, but in the middle, very apparent, is that hemorrhage, that blood hemorrhage. That is the aging form of macular degeneration. About a decade ago, the first genes were identified that contribute to AMD, but there's a real difference. This condition, Stargardt maculopathy, that's one gene causing the problem. Here, for age-related macular degeneration, there are about 50 genes that contribute to the problem, and so you can immediately understand the difficulty in sorting all this out. So tonight, I'm going to focus on the single gene problems, and particularly at the back of the eye. Now, this work got a big boost 15 years ago with the completion of the Human Genome Project by Francis Collins and many, many others. And I would say it's a seminal turning point for understanding disease. A gene gets you into the process of disease and into the processes of cells, and it is literally leading to medical breakthroughs. Now, some of the tools that you may hear about or read about in the local papers or the New York Times, genetic diagnostics. That really is possible courtesy of the Genome Project. Gene therapy, we're going to talk about that in a moment. And other ways of employing genes that are making a fundamental difference in medicine already, and certainly more in the future. Well, the litany here is genes go to cells, go to understanding disease mechanisms, go to treatment. So that's what we'd like to see happen. And ultimately, you can follow the pathway down. And one of the pathways that I'm going to follow tonight will be gene-based therapies. there are many other things, obviously, you can do therapeutically when you know the genes responsible. This is a story that you have been reading about in the papers just the last 6 months. The story goes back to Dr. Theodore Leber a century and a half ago. These stories really started very early because Dr. Leber looked in the eye of a child and saw pigment spots and disturbance. Here you can see the pigment spots at the edge. It means that there's an injury to that retina, that nerve tissue. Dr. Leber put together the story of looking in the eye and seeing a problem with the child presenting who had no vision, severe vision impairment, or even total loss of vision. No family history because it's a recessive condition. And characteristically, the eyes would be bobbing around, which if you see someone on the street with eyes bobbing, you know they're not looking at you because they can't see you. That's why the eyes are bobbing. So this story started a long time ago, and then about 140 years later, 1993, Dr. Michael Redmond, a scientist who happened to be at the National Eye Institute, identified a gene. He didn't know what the gene was doing because this was before we had this burst of genetic information over the past 15 years. But through some scientific sleuthing, he finally figured out that it was a very abundant protein in the retina. What do you do with it at that point? Well, you eliminate the gene from the retina, and you do it in a mouse. You genetically engineer a KO, a knockout of the gene, and then you ask the mouse, how well does the mouse see? That's a tricky question to ask a mouse, but you can, in fact, do it. And then, quite surprising, a dog turned up with a mutation in the same gene. Now, that's important because this is a naturally occurring mutation, whereas the mouse is a laboratory mutation, so you can begin to understand the complexities of this. But the dog was particularly interesting because several very astute scientists, including Bill Hauswirth and Gene Bennett, and many others, did gene therapy on the dog. What does that mean? It means that they put the proper gene in, the RPE65 gene, into the dog's eye, and the gene was incorporated into the fabric, the structure of the eye, the biology of the eye, and it restored vision. And in a dog, you can tell right away if the dog has or doesn't have vision. There is an important laboratory test called an electroretinogram. That's what these wavy lines are here. In black, pre-treatment, so before the treatment was done, there's not much activity. And after the treatment, post-treatment, you can see that there's a lot of activity. Now, just for reference, the way you measure this activity is you put a contact lens on the eye with a little wire ring on it, flash a light, and the eye responds with an electrical impulse, the way you do an electrocardiogram. Very similar, but this is for the eye, electroretinogram. And with very dim lights, not much happens, but as the light intensity increases, you can see that the responses build up. So this is a good way to check a mouse, for instance, who can't tell you, I see or don't see. If you hear a mouse talking to you, I've got some recommendations of people you can see. So this was successful in the dog in 2001, not that long ago. And then very quickly, this went to human trial. Here you see Al McGuire in surgical garb, about to put the gene into the eye, a gene in solution. They put the solution in the eye, it carries the gene. He is treating this child here. One of the nice things about the area of vision research is it's a very rich resource of people. And there were actually 3 different groups simultaneously in 2008 it was, here's 2008, that did gene therapy for RPE65. I'm going to show you a video, if you could play this video. This is a video of one person, before treatment and after treatment. So when it comes up, you will see that the one person is walking quickly through the maze. But her former self, before treatment, if you could play that, please. Her former self, here she is. Look at her after treatment, just walking right through that maze. And here before treatment, she's really struggling with what's happening. The lighting is turned down in these pictures because this is done in dim light. That's the most sensitive way to test for vision. And before treatment, she is still finding her way. She can see, you get a sense she's following this, but not very well. Whereas after treatment, she's already done. She's just walked right through that, which gives you a sense of the value of this treatment for these individuals. Very few of them actually in this country, but these individuals who have lost their vision courtesy of being born with a mutation in the RPE65 gene. I'm going to move on now to the next slide because the punchline on this is that the treatment has now reached approval by the Food and Drug Administration. So FDA drug registration, this occurred just several months ago in December 25th, 2017. It has the name Luxturna. This is the FDA statement. Luxturna is the first gene therapy approved in the U.S. to target a disease caused by mutations in a specific gene. So as director of the Eye Institute, I am pleased to tell you that vision is really poking along great. Research in the area of vision science is front and center in many of these novel therapies. One of the interesting questions that we're not going to consider tonight is how much should you charge? What does it cost? What's the value of taking a person who's blind and giving them some vision? Obviously, the company is Spark Therapeutics, has its version of it, and others have other versions of what the cost should be. So that's a quick story. It did start in 1850 with identifying the problem, and 1992 and 3 with finding the gene, and 2001 with treating the dog, and now 2017 with an approved treatment that you can get at your pharmacy, figuratively said. But this is not the only ocular gene therapy that's going on. Here is a table, a list of many of the targets that are currently in clinical trial. And right at the top is RPE65, Dr. Theodore Leber, congenital, you know what that means, born with it. Amaurosis means no vision, Leber congenital amaurosis, or LCA. This is now at the stage, has received FDA approvals. These others are in the clinics. And one that I'm going to spend much of the remaining time on is called X-linked retinoschisis. The trial that I am doing happens to be using this virus. There is a company that started, I'm pleased to say, a little bit after we began our work at the National Eye Institute. And I'll explain in a moment what AAV means. But before we go on to that particular trial, let me just orient you to where all of this is happening in the eye. You recognize an eye, at least from a biology textbook, that's what it looks like. And at the back of the eye, the lining on the back of the eye, inside the back of the eye, is a layer of nerve tissue called the retina. And so this little cutout here is expanded to show a cartoon of retina. And this cartoon is further expanded on your right side to show the rods and cones. The cones come in red, green, and blue, so that's why those colors are there. Those are the light detectors that initiate vision. You're looking with your rods and cones right now. When it gets very dark in your rooms tonight with the lights turned out, you'll be using the rods because they're very sensitive. So it's the rods for darkness and the rods and the cones for brighter light. It turns out that all of the conditions, I'm going to go back a slide, all of the conditions here involve those rods and cones and retina tissue. It's a tissue that is susceptible to genetic diseases. So let me tell you the story that I've been involved with, which is called X-linked retinoschisis. This one also started more than a century ago when Dr. Joseph Haas identified two brothers who had unusual cavities within the retina when he looked in. And those cavities kind of radiated out in a spoke wheel fashion. And so he described these, and someone drew what he described as the image that he was looking at. He happened to be an Austrian ophthalmologist. There were two brothers. That's important because the X chromosome is critical for making us poor, weak men more susceptible to disease, the X chromosome. And so this gets the name X-linked, means it's on the X chromosome. And the retinoschisis are those cavities. And it comes with poor acuity. Here's a picture of the equivalent of an 8-year-old boy that I had seen at some point who had difficulty reading. He was kind of clumsy if you gave him vision tasks to do. He didn't do fine things very well. He failed the vision testing at school, went to an ophthalmology office, and the doctor saw these radial spoke wheel cavities, just like Dr. Joseph Haas did. And he gets the diagnosis of retinoschisis. He's got poor acuity. Now, over the past 20-some years, there's been an important new tool in ophthalmology offices, in vision testing offices, called the optical coherence tomography. It's kind of like ultrasound. You bounce waves off the tissue, in this case the retina tissue, and you see the structure of the tissue with tomography. Here is what everyone in the room would have, or at least I'm presuming most people have good vision. Be looking at the slides, and you have this dip for the fovea. Here's your fine vision for reading the LA Times. There are these rods and cones down here, the photoreceptors. They catch the light, photoreceptors. But look in the retinoschisis retina, the same image, look how disturbed and disorganized that is. And here are these cavities. Those are those radial cavities. So you can actually see the structure of the retina, and presumably if you could do gene therapy to fix the retina, those cavities would disappear, and you could do your OCT in the doctor's office and just look. So that's the center of vision, the fovea, the macula, the center. In the periphery, there's a different kind of havoc going on, and that is that the surface of the retina is just kind of tearing away, like you're pulling tissue paper away from the underlying nerve tissue. And so these eyes are susceptible to the equivalent of a retinal detachment. Not a good thing to have, not a good thing to live with. Here are the cavities, the radial cavities, and the cavities on OCT. There's one more tool that I want to introduce, and that was the electroretinogram that I showed you for the dog. If you happen to have great memory and recall those ERGs in the dog after treatment, there was a wave that went down and then went up. That's what a normal would look like. When you do the test, it's an office test, when you do the test on men who have retinoschisis, it goes down, it just doesn't come up. And one would presume that if you could treat the retina, you would convert this abnormal functional response to this more normal-looking functional response. So here on this slide, you kind of capture some of the important elements of this condition. Obviously, poor vision, central radial cavities that you can see the structure of, and on the bottom, an indication that the nerve cells are not, retina nerve cells are not working properly. Okay, so that's where knowledge stood until about the year 2000, actually 1999-98, when the gene was identified, the gene that causes this problem. In shorthand, it is RS1. That's the gene. And if you knock out the gene, the KO, you can do that in a mouse in the laboratory and give the mouse the human disease, or at least that's what you hope for. So let's see if that works here. Here's the gene, and we manipulated the gene to disable it. We look at protein, and there's no band here. Here are two bands, but no band, and it says there's no protein being made. That's good, because you knocked out the gene. And then you cut the retina in slices and look at the photoreceptors. This is in a normal animal. You didn't do anything to this wild type WT mouse, normal mouse. This is an antibody labeled in red, and you can see the protein is here and maybe a little bit here. But after you knock out the gene, the protein's gone. Plus, it's kind of disorganized. You've got some structural problems. Then you record the ERG, electroretinogram, in the mouse, and bingo, you've got the functional equivalent. Here is the normal. Dim lights, not much activity. Brighter lights grows to a normal response. But in the mouse, the knockout mouse, that upgoing wave called the B wave is missing. And here are three examples shown with the red arrows. So you've got structural correlate and functional correlate in the animal. That means you can begin to use this animal to really study how to develop a therapy. And this is the same thing that's being done on all major campuses now, UC Irvine and Stanford and UCLA and even, I shouldn't say that, Michigan. Well, I guess after the other night we should treat them carefully, shouldn't we? What's the story with this protein? You can't just go in and do gene therapy on this mouse. You've got to begin to understand what you're trying to accomplish. And the question is, what's the protein? What's it doing? Insulin, for instance, is a protein. What does insulin do? It regulates blood sugar. What does this protein do? Well, let's try and find out. I've shown you now 2 or so slides that look equivalent to this. This is a section through the retina. These are the photoreceptors in red, and the cell bodies of the photoreceptors are in blue. So these are the light-sensitive cells, photoreceptors. Yep, there's plenty of protein there. And then the synapse zone between the photoreceptors and the second set of cells, the second-order cells, there's a lot of protein there. Those are the synapses. So those would be fruitful areas to begin to understand what this protein is doing. And I'm going to show you both of these. I'm going to start right here with the segment of the photoreceptor, and then afterwards we'll look at the at the synapse. But look, first, let's look at those photoreceptors. These are electron microscope pictures of the photoreceptors in a normal animal. And the little black dots are labeled, they're immunogold, they're gold particles that target the protein, attached to the protein. And so the gold particles tell you where the protein is, and there's lots of it here. There's none here. Here in another view, there's not much, there's really none there, but there is here. This is called the inner part of the light-sensitive element, the outer part, the inner segment and the outer segment. And very clearly, this protein lines, kind of paints, it coats the outer membrane of the inner segment. And where you look two inner segments side by side, you can even see that the protein is right there between the cells. So now you at least know where it is in the cell. But after a heck of a lot of work to get these pictures, you still really don't understand what's going on. So we decided we would look at the protein itself. And that turned out to be quite an adventure because this is a brand new tool. It's an EM, electron microscope picture, but we're taking a picture of the protein. And the way you do that is to take very pure protein, embed it in ultra-thin ice at ultra-low temperature, and you have an ice film with protein embedded. And sure enough, here you can see the individual, can you see those? That's actually the protein you're looking at, the thing we've been working for a long time to understand, gene cloned in 1999. And if you have a good imagination, you can begin to see that it's an 8-pointed star. Well, instead of imagination, what you can do is cut out these pictures and optically overlay them and average them together to improve the clarity. And when you do that, in fact, here, yep, 8-pointed star. So there are 8 of these proteins. They're in a football huddle or whatever it is. And kind of flat like a frisbee, but then amazing to us, here's one set of 8 in a frisbee, and here's a second set of 8 in a frisbee. These things are all tightly organized. Now we're seeing that there are 16 proteins all tucked together. Well, this is with 300 pictures averaged, so we said, don't let a good opportunity go to waste. We averaged 11,000 of them. And then you see very interesting things. If you could play this at the bottom of the image, please. Yep, let's see what this looks like. So there you see 16 retinoschisin proteins, all nicely tucked and packed together. And there are bridging elements that hold them together, and the tails, here's one, and the tail ends up in here, and here's the neighbor, and the tail ends up in there. So you've got 8 tails that are all interweaved. Amazing. And then you've got to start imagining, what in the world could this be? Well, here's one way to imagine what's happening. If you remember, the immunogold EM showed those black dot particles between 2 cells lined up there. That would be the equivalent of this. Here is cell 1 at the top, membrane of cell 1, membrane of cell 2, and the 16 molecules, the 2 frisbees, are sitting right next together. Here is one frisbee embedded against the membrane. Here is a second frisbee embedded against the membrane, and when it gets close enough, they stick together like Velcro. Perhaps you can detect my skepticism on this because I labeled it perhaps. There are a couple problems with this, but boy, it was fun to think about it while it lasted. Okay. All right, so now let's look at that second region, the region of the synapse itself. And you can't really see much here, except you can see that in the synaptic region there are a lot of these gold particles that are targeting retinoschisis protein. So you know the protein is sitting right there in the synapse. You knew that previously, but this is real proof of that. Now on the top is the photoreceptor cell, the light-sensitive cell, and on the bottom is the second cell. And the photoreceptor catches light, but then it's got to plug it in for the vision to get to the brain. And that plug is this synapse, the junction between the two cells. So, sorry to bother you with some real details, but we've got to think about how a synapse works for a moment. On the top is a photoreceptor cell, and on the bottom is the second cell, neighbor cell, the bipolar cell. And the first cell catches light and releases a transmitter, which is called glutamate. That glutamate ends up being picked up by the second cell, the receptor on the second cell, and telling the second cell that the photoreceptor saw something. Now, the details, and this is important because the next two slides are going to show you the real-life molecules. This is a complicated synapse on the receiving end, the postsynaptic side. There's a receptor, and then there's a channel that tells the cell to do something. And then there's some shuttle molecules or cascade signaling molecules, number 2, the signaling molecules that communicate between those two things. So the next slide will show you the receptor and the channel on the membrane. On the top, the green dots, wild type, normal animal, and the knockout animal, plenty of green dots, says that there's plenty of receptor there. That's not the problem. The receptor's there. But the channels, these little tiny green dots, yep, they're there in the normal animal, but they're missing in the knockout animal. So that's where the problem is. If you can put those channels back, you will have fixed the problem, we think. And then what about those shuttle molecules, those signaling molecules? Well, here you can actually see the benefit of gene therapy. On the top, the untreated animal. On the bottom, the treated animal after gene therapy. And after therapy, all the proteins are nicely localized in those little tiny dots. But on the top, they're just scattered all over the place. So they're ineffective. So what this says is a couple of things. First, that it localizes the vision problem not to the cavities that are there, but to the synapse isn't working. So this is a synaptic disease. Second, it says with this particular gene vector construct, you can repair the problem. So presumably, if it works in a mouse, somehow we're going to get this to work in human. And I'll just slip over this quickly, but instead of a Velcro model, this I think is more reasonable. Here is one cell and a second cell and the space between the cells and the retinoschisis, two frisbees in blue sitting there, and it's holding some of these other molecules in place. It's a coordinator. It's an organizer. That much I think we're pretty sure of. Exactly what it's coordinating, whether it's the receptor, well, probably not, because that didn't get affected by the knockout, but it certainly does affect the localization of the TRP channel. Does it work? Yes, it works. Here's an animal treated in one eye, the other eye not treated. This is one of those mice where we've done the gene manipulation. And in the untreated animal, again with this ultrasound type imaging, you can see the cavities here. You see those? Compared to the nice, tight, closed retina after you've done gene therapy. So you fix the structure. And here, in green, that positive B wave came back. So this vector fixes both the structure and the function. Wonderful. I'm sure all the mice in the world are happy about this. But is it going to work for human disease? Well, that's the next big question. So we're doing that. We started our trial in 2015, and I'll show you just a couple of slides to tell you where we stand at the moment. To get to trial, the key elements to get to trial are to show that this vector is not toxic, that it's safe, and the second, that it does something. Well, the last slide, this slide says it does something, and then we did the safety studies in a lot of mice and rabbits, and ultimately went to the FDA. Just so that you can really understand a little bit of the nuts and bolts of what gene therapy means, let's talk about what this vector is. The vector has three parts. Very, very simple concept. The vector has a virus. Viruses invade tissues. When you get pneumonia, it invades the lung tissue. So viruses like to get inside cells. And in this case, you ask the virus to pull along with it the gene that you want, retinoschisis gene, and the promoter, kind of the street address, the phone book. You've got a promoter that works in photoreceptor cells and a promoter that works in heart cells and a different promoter works in kidney cells, etc. So this is the gene therapy construct: a virus, a gene, and the street address, the promoter. And here's our virus. We're using an AAV type 8. Here's the gene, RS again, for retinoschisis, and the promoter. So it's a standard kind of gene construct. How do you deliver this? I keep talking about we delivered it. How do you deliver it? Well, you need to get to the affected tissue, which, if you recall, are the rod and cone tissue at the back, the retina, nerve tissue, complex tissue, very organized tissue. And if you are real clever, you can stick a tiny needle underneath the retina and put in some of the fluid that has the vector. So that's this little green bubble, subretina, underneath the retina. There's a problem with that. You've treated only that one little area. You haven't treated the rest of the vision. You've treated one area of vision. The approach we took was to just put the fluid into the empty space in the eye and let it diffuse where it's going to go. There are some biological, technical reasons why this shouldn't work. So we think it will, but there are some cogent arguments against that. And other therapies at the moment are using this local subretinal delivery, but that's how we are doing it. Now, this is the last technical slide. This shows the kind of data. FDA, you walk into FDA and you say, we want to do a gene therapy experiment. And they say, well, could you give us some information about what you plan? And so this is the information you give to the FDA. Not hard to understand, actually. There are three ideas. One on the left, one in the middle, one on the right. The one on the right is called structure. If you remember those cavities, if you close the cavities, the retina ought to get thinner. If you've got cavities, the retina ought to be thick. And lo and behold, with low dose, here's the thickness, but with increasing dose, the retina gets thinner. It says the cavities are disappearing. It's a quantitative way to say that. Here is how much protein is being made. That's how red the tissue was if you use that red antibody. Low doses, not very much redness, not very much protein. 10 times higher, 100 times higher, you can see that the protein expression goes up. So there's protein that's been made there by the vector. And the function, nothing happens, nothing happens, nothing happens. Oh, there it goes. Now it's working. And if you can read the fine print on this, I've circled in red 1 times 10 to the 8. 10 to the 6 is a million, so that's 100 million virus particles that we put into a mouse eye. 10 to the 8, and it jumps up at 10 to the 8, and the cavities close at 10 to the 8. So we put the protein in, and it's done what we expect. And FDA said, bless you, go try it. Well, it costs us a few months and more of work. This is part of the application we put in, 24 inches of documents, mostly data, because they want to know safety and does it work. So that's the kind of application you put in. There's one more thing you got to do. What about the patients? Where do you find patients? Well, we had been busy at the National Eye Institute in seeing many retinoschisis patients for a number of years. And we study them so that we can understand the disease and the implications for their lives, their vision. And this is interesting because at age 0 or 20 years of age, 40, 60 years old, 80, nobody was 100. And this is the thickness of the retina, very thick here on top, 800 microns. Normal is about 250, that's this dashed line, and this is a thin retina. So at younger age, up to about age 40, the retina has a lot of these bubbles in it, those cavities, and the retina is thick. And so these are above the line. And then the natural history of the disease, the natural course of the disease, is for those cavities to close, and the retina gets thin. So if you're going to use cavity closure as your measure of success, you would not want to have people at age 60 and 70 because they don't have cavities. You'd want to have people 40 or 30. When you get to 10 and 12, that's an age that you want to know you really have a good chance of getting success before you invoke younger people. So we've been working with individuals from 20 to about 50. How much do you put in? Well, 10 to the 8 in mouse. The mouse eye is a tiny little eye. How do you scale that tiny mouse eye up to a full-size human eye? And you can do the geometry. I relearned my high school geometry here. And instead of, here we went 10 times higher. Now we're 10 to the 9, 10 to the 10, 10 to the 11. Those are big numbers. 10 to the 9 is a billion virus particles in the eye. Turned out, no problem. The immune system of the eye handled it fine. 10 to the 10 started to be on the edge. 10 to the 11, the immune system is doing its surveillance and causing inflammation. Fortunately, it's self-limited. So those were the original doses. We've now gone through those doses and we're on to other doses. What are you going to look at? Well, that ultrasound, actually called optical coherence tomography, you're going to look at the cavities, you're going to look at the electroretinogram responses. You're going to ask the person, do you see better? You walk the maze better. There are a lot of ways to monitor vision. And then you're going to look at some serum chemistry. So does it work? Well, we've got some indication that's hopeful for us and for the patients, subjects. Here is an OCT image and the cavities right in the macula. You can see them. And within weeks of administering that vector into the vitreous, those cavities had closed. We've only done a limited number of people so far, so we're going to need to repeat this and at the same time be monitoring the acuity and visual performance, visual function very carefully to know whether this works. But I've got my fingers crossed, but I'm hopeful that in fact we're seeing some signs of success. This is essentially the last slide. Let me just wrap this up then. Gene therapy, simple idea. Gene's broken, fix it. Well, it does take a little bit of work to get there. I would say, in the least, it's still highly experimental. This is not ready for prime time in medicine, but it's going to be within the next 10 years. I talked about the immune system. The immune system, when you put these virus particles in the eye, the immune system pays attention. Of course, the immune system keeps us alive until age 93, so it's doing its job. And we're going to need to be smarter about the kinds of viruses we use to make them more potent to deliver, but less excitable for the immune system. And at the moment, there literally is this one successful gene therapy across all of medicine, and it's for that childhood blindness, the congenital amaurosis, approved in December 2017. And as I said, how do you price that? That's critical because this is not inexpensive development work, but it is fundamental, I think, in the future for medicine going forward. And more than anything, I think at this point, as perhaps I've tried to do in this talk, is to manage expectations. Because if you walk out on the street and you tell somebody, yeah, we're doing gene therapy, they say, oh, can I have it? No, not yet. We've got a little development work to do. This is not magical medicine. This is good old-fashioned, hardcore research that's happening in 2018. So I hope I've interested you. I presume no one in the audience has retinoschisis, but if you do, I'd sure like to talk with you. And just to know who you are, too, because it looks like a nice group of people. So I had a lot of help to do this. We've got a good group of people. I run a laboratory. Those are the names on top. Some other people at NEI, National Eye Institute, developed the vectors. And on the bottom, we have dedicated clinicians who are working through this whole story. And here's the NIH where I work. But maybe I should go to UC Irvine. I think it's a pretty good place. Okay, good. Thank you.